Thank you very much, Chair. Good evening to all of you. I would like to give you a short presentation. It's not a long presentation. So this is my title of presentation today. Harnessing microbial diversity in culture collection. So yesterday I talked that we have a culture collection in Indonesia, and thank Dr. Suzuki Sensei and also our Sensei for establishing culture collection. And now we talk about our projects, a current project with NEDO. So new energy and technology innovation of organizations, like development and organization. This is the project is different from our previous satellite project. It's focused on energy. So I will talk about five main things. The first is brain organization. So we have a new organizations now, and then we talk about net zero emission program. This is related with net programs, and then biohydrogen, lipids, and surfactant productions. I think a few years ago, we have problem on how we can manage the international budget, especially how we can be more efficient. And therefore, the government merged two research centers. One is LIFI, that's the, and then BPPT, Agency for Assessment and Application Technology, and National Nuclear Energy Research Centers, LAPAN, so very big research agency. So we have about more than 10,000 scientists in one research agency. You can imagine how to organize that very big research center. The objective is centralization and streamlining funding, resources, and policy. So then, and then of course the strategic priority focus on national challenges, development, health. Then one of the program is the energy program. Yep. I think we have in our research center, so this is the before. This is the brain, so we have more than 5,000 scientists, and the culture collection is here. And this was LIFI here, okay? And our research center is Natural Science and Environment under this organization, so a very big research agency. And then our research focus on three major things. The first is the enzyme technology, cell microbial technology, and fermentation. This is under the Research Center for Applied Microbiology, with myself involved in these projects. Then also we have many research groups, and I belong to the this one, the energy section. So this is our project now, ongoing projects. The innovation in polymer wastewater treatment technology using an anaerobic facultative anaerobic system. So we use an combined anaerobic and aerobic system. I think Dr. Lee very familiar with this system. With superior biohydrogen processing microbes. So biohydrogen, I think one of the satellite project with Japan is now dealing with biohydrogen productions with microalgae, but we look at for different projects. So why this is important? I think you look at to Indonesia, we have quite large amount of palm oil. So we have about 395 palm oil mills spread across Indonesia. And the production volume is more than 50 million ton per year. And this is possible, if we can compare. It's main big problem in Indonesia. Possibly we can produce what? Hydrogens. Before we just produced methane, but now if you modify the treatment plants, you can produce biohydrogen instead of methanes. So for this project, we involved several research centers involved in these projects. We tried 3 stages of fermentation. So dark fermentation, photo fermentation, and 2-stage process. And in this process, we work together with France for development of continuous process for biohydrogen productions. That's when we do parallel research, not only in the lab, but also try directly on treatment plants. So this is our treatment plant in Riau. how we can produce biohydrogen together with methanes. So for this research, we do a collaboration between Japan, Indonesia under the grant obtained from NEDO. So we work together with University of Tokyo and also Kobe University. It's very difficult to define the word, which work should be conducted in Indonesia and which could be conducted in Japan. But through this project, we try to work together. So our work is here. This is mainly conducted in Indonesia. So we have treatment plants in Indonesia, collect the POME, and we do analysis, total analysis here about using GCMS, what kind of component it is, and then we try to stimulate what kind of process is more efficient to produce biohydrogens. So we use a system, anaerobic system, to produce not only methane, but also how we can also live it. So we do biorefinery projects in this case. Then with those three technology. So now, we obtained already several superior bacteria for producing biohydrogens. We do laboratory tests in Indonesia. This is all of my work to do sampling, and this is the use optimization processes, so how we can do an anaerobic system to produce biohydrogen and methanes. Of course, when we have a bacteria, we have to study them and what kind of enzyme involves. So University of Tokyo working in this direction. So when we have a glucose and how glucose could be effectively converted into hydrogens. For the moment, for this year, we have 2 patents. One on the system of facultative anaerobic bacteria for biohydrogen production, and one is lipid production system using POME. So, and we published one article on statistical optimization of culture media components for enhanced production of lipids by yeast. So the yeast is a collection of Kawasaki Sensei. Okay, so next, so again, it's only going to Russian word. Okay, what? Okay, now, if we look at, we already isolated several bacteria, but how is the process of producing biohydrogen can be, per moment we produce 1.2 mole. In fact, theoretically, from 1 mole glucose, you will produce 6 mole hydrogens, but we produce only 1.2 mole. Therefore, we have to optimize the process, so especially how we can do metabolic engineering. So how we can increase the biohydrogen production through this system. So from glycolysis, so optimize the glycase process here, and then from this glycolysis to they are through several pathways. One the pathway here is to pyruvate and acetic to acidogenesis. So this they produce more hydrogens. So the University of Tokyo working on the hydrogenase system, how they can optimize the conversion of glucose into hydrogens. It doesn't work. Okay, so after knowing this, then we have several microbes. But what kind of microbe is the best for these processes? From the theoretical point of view, if we have 1 small glucose, we'll have 12 hydrogens. Is that right? But in fact, it's very difficult to get it. So we modify now the system. How long is aerobic condition, how long anaerobic conditions, and what is the FM ratio, the food for microbe ratio, and many others. We are doing consortia or single culture or consortia cultures. Okay, sorry, thank you. Now, I think yesterday many people were talking about how we can combine a microbe. First, we isolate Acinetobacter radioensis, Cosaconea, and Cosaconea species. And now the idea is how we can combine this bacteria with Lipomycin one very important lipid accumulating yeast, usually in other startup projects. What's happening is now is that can we grow together? Can we grow together? And what kind of medium is good, best for them? Yeah. Then we do compatibility tests, how we can grow better yeast and bacteria. So we do compatibility tests. Of course, we do with several media, we can media, and also incubation temperature, pH, etc. Next. And then we test. We do co-culture, we can single cultures, and then how we do dragon productions. Whether they can increase the difference between single culture and co-cultures. So how is, and then next. So and also we study the pathway. So we do a test for do LCMS. When we do single culture, what kind of metabolite produce it? When we do co-culture, what kind of metabolite? And try to understand the pathway. I think next. This is the pathway. Next. Then we test the hydrogen production. What kind of gas they produce it? How much methane? How much the other gases? Okay, of course we use several media. Try to use glucose, POME, combination, manifold dissolution ratio, FM ratio, etc. And then next, and then we do study also again the pathway. Yeah, the question is that when is best? When is lipid now not only hydrogen but also lipid? So in POME, can we use lipid, hydrogens, and other substances? So we study them. Of course, we have studied the pathway. Next. Okay, next. Again, we do LCMS study and GCMS study. Look at the pathway. Next. Okay, next again, then we combine cocasonia and lipomyces. What kind of product they produce it? Of course, also we study what kind of lipid they produce it using FTIR. And then finally, next, lipomycin if we do single culture, combined cultures. Next again, we also use the acidobaradiensis. Next. Okay, again. Okay, next again. So many data. Okay, next. Okay, finally. So I studied facultative anaerobic bacteria from 1997, so about 25 years ago. And finally, now we try to, we got now the technology how we can produce biohydrogen in POME. Of course, under collaboration with Tokyo University and other research centers. And we have now 2 patents and 1 paper on high impact journal. Next. Next step is that when we produce, stop there. Okay, so when we already got the bacteria, how we can use it in the fields? I think he's a very famous professor, I think, Professor Chiagi Ogino from Kobe University, and he has a lot of projects in Indonesia. Now, if we have that culture, can we do also metabolic engineering? He is very interested in that. Co-culture is a bit difficult because it's very difficult to maintain the cultures. But if we have superior microbe with superior capability, of course, we're using metabolic engineering. Can we use it in the future? Okay. Then, sir, thank you. Next, it doesn't work. Okay. We got several bacteria, very important bacteria. Here is AC21 and AC23. This bacteria are very strong on biohydrogen production. They have amylase, cellulase, protease, but not with lipase. But this bacteria has lipase capability. But if we combine this bacteria with this bacteria, what will happen? So we try to combine this bacteria with automation, okay? Of course, to do that, also we have to understand how is the biohydrogens production system in each isolate. We have to understand that the Nojiri Sensei, one of the professor in the University of Tokyo, helping us on that verification of the heterogeneous system of the isolates. Then next again. Oh, okay, finally we got very good result on this. On this combination of AC21 and AC23, we have very pure hydrogens. And the bacillus is Acinetobacteriensis, Bacillus cereus, Weisela paracinolis, Enterobacter. So this kind of bacteria is good for biohydrogen production. Next. So, okay, next again. So this conclusion, the Acinetobacteriensis and Coxsackia species are good for hydrogen productions. And also, of course, if we can modify the medium, putting glucose, also lipid will be produced. Time is limited. Is enough already? It's finished. Okay. Then conclusions is in this bacteria strain capable of biohydrogen production successfully isolated from our our sample, and scaling up innovation. This virus and reactor design, process control, with anodization system. Of course, we will work together with the Japanese scientists, and hopefully also we can get both of us, of the collaborator, get synergistic benefit. I think the strength will be the partnership leveraged in local Japan bioengineering system. I think that will be important for us in the future. The future challenges will be how we can optimize the hydrolysis and also to scale up the production. I think that's my presentation. Thank you very much for your kind attention. Thank you. I think University of Tokyo, nice, yeah. And I think we have quite a lot already, but I think this is my main presentations. I don't want to keep you sleepy. And thank you very much again.